Like
Ciao,
I-
ciao, ciao Pedro.
it's good seeing you. Okay, I'd like to keep on time. I'd like to say that I can summarize because all the stuff that Paolo has said, I'd like to summarize my knowledge of him in 3 moments. And it's just one of the aspects that he does. I remember many years ago, I saw this guy who was the president of the International Federation of Clinical Neurophysiology, writing an editorial on neuroimaging, multimodal imaging. And that struck me as very interesting, very advanced for the time he wrote it. We later talked a bit about this in the Congress in Uruguay. Then many years later, after we held in 2016 a meeting of the big brain projects in Geneva with WHO, we also contacted Paolo, and we discussed about how clinical neurophysiology could be useful. And I learned of his ideas of precisely using of creating a front end for the public health system, which would use neurophysiology and to carry out these trials in Italy and in other places. Then finally, I have to say that just very recently, we met a few months ago, and we've been discussing all this time on the real role that neurophysiology can play. And I have to say that in spite of being one of the most eminent people in the field, no, one sometimes associates with people that work at very specialized hospitals, I have rarely seen the interest in having our specialty, which is clinical neurophysiology, actually be an important part of public health and of changing health indicators. So this fits perfectly well with the mandate that the GBC was given on trying to have an impact on the global burden of brain disorders. Now, due to all this, most recently, Paolo was named as the liaison between the International Federation of Clinical Neurophysiology and the GBC. And we asked him, and he honored us by becoming one of the members of the steering committee of the Global Brain Consortium. So everything else you can read on his CV. I just wanted to give some personal impressions of how and how thrilled I am that there's a person with this level of knowledge in neurology, clinical neurophysiology, that at the same time understands what the population needs and is trying to do very seriously work in this direction. So, Paolo, thank you much once again for joining us.
Thank you, Pedro. It is my honor. And I am ready to give my lecture if you think that it is the time to do it.
Yes, it's now the time exactly, okay?
Okay, now I'm starting my presentation, first of all, thanking the organization and in particular the Global Brain Consortium and Professor Pedro Antonio Valdesosa for inviting me. I am Paolo Maria Rossini. I am an old neurologist, more than 70, already retired officially, but still working a lot more than I was young. And I see patients, so I am a clinician, but my whole research life was devoted to clinical neurophysiology. And I will try in the next few minutes to convince you that today clinical neurophysiology and different techniques of clinical neurophysiology can provide useful information for the early diagnosis of a disease, which is probably the real challenge for the next decades, which is the cognitive decline linked to neurodegeneration and to dementia. Let me recall myself and all of you who are not real experts that we record our EEG signals, electroencephalography signals through scalp electrodes. So this is a non-invasive method that we rely on the basic signals are represented by the rhythms of the EEG. So the oscillations of the EEG in the different frequency ranges spanning from the delta to the gamma, which is the fast one. That today this method is really portable, wireless, and can be performed at home, and the signals can be put on the web and analyzed by an expert center everywhere in the world. And finally, that since its origin, these are figures taken from the recording of a one-channel system in Harvard in 1934, that EEG signal is quite sensitive to changes in the mental activity. So if the brain is at rest or is solving a mathematical problem, the rhythms change immediately, and they reflect somehow the mental activity and emotional activity which is taken simultaneously place in that brain. So we can record from now not only one channel, but several channels, up to several hundred channels in a resting state condition. During sleep, which is very important, is a completely different story. There is no time today, but it is quite important to consider also the sleep characteristic in neurodegenerative disease, or during a task, when the subject is processing something and is providing a response, correct or wrong as it is. And the second and final part of my presentation will deal with some types of transcranial stimulation, the magnetic one or the low current one. The high current intensity is no more utilized because it is painful, and it belongs to the pioneering ages of transcranial stimulation. Since the introduction of the first microprocessors, it was possible to demonstrate that the computer analysis of a complex signal, as it is the EEG one, can be quite helpful. A simple demonstration is that in an EEG signal, we have the overlap of the different frequencies that we have spoken before, that it is not possible to discriminate them by eye, but it is possible with a simple Fourier analysis to see quite nicely and to measure quite precisely the contribution of the delta, of the theta, of the alpha, of the beta, and the gamma rhythms. It is also possible today to make a sort of tomographic scan of the electroencephalographic sources and to analyze the regions of interest. There is a quite popular method available for free on the web, which has been carried out by Professor Pasquale Marquis in Switzerland, in which it is possible to discriminate the contribution of frontal, central, parietal, occipital, and temporal and limbic areas, at least. You can increase the number of regions of interest by increasing the number of recording electrons, but this is also another condition. It is known that the brain has a local specialized area, but really this is an old view. In some respect, every area contributes to most of the brain activities in a different way and in a different percentage. So there are areas mainly devoted to sensory processing, but they also contribute to memory and to emotions and to other cognitive activities. So the complex task requires the cooperation between multiple brain areas, cooperation that can be in series, so in parallel, in series or in parallel, so with different temporal contribution. That the rhythmic firing synchronization is one of the key mechanisms of functional integration, is one of the brain languages. So if two areas behind these two electrodes are speaking each other and there is a flow of information, For example, you may see temporal transient changes in the coherence, in the synchronization likelihood of these two oscillating activities at these two electrodes, and they go in phase and out of phase in few tens of milliseconds, providing us a measurement of how much is the flow of information between these two areas. Consider the time discrimination, because the millisecond time is something that cannot be reached by any other technique dealing with the brain activity, including a functional MRI or PET. So when synchronization of two different brain areas is repeated in time, this leads to the formation of functional network, which have temporal and spatial structure. At the very end, we can consider the brain is one of the model of the functioning brain as a place containing a number of oscillators that go sometimes in phase and very often in out of phase. So they are working each other through a fragile type of binding, unbinding. And there are pathological conditions in which this mechanism is not working anymore. For example, when there is an excessive type of binding, you may stay within an epileptic focus, or in a place where neurons are producing dyskinesias, or in a place where there was a damage to the brain tissue, like a stroke. On the contrary, on the opposite side, when there is a decreased amount of capacity of binding, synchronizing, going coherence, in all the pathologies producing disconnection. And this is the field of the neurodegenerative diseases which I'm speaking about today. It is important also to consider that the neurodegeneration, for example, Alzheimer's type dementia, but this is true for all the neurodegenerative disease, do have a prolonged time during which there are no symptoms. So a pre-symptomatic stage in which there is only one functional problem, which is usually at the level of the synaptic transmission. And this may take years. And after a while, there are the deposition of toxic agent, for example, amyloid in plaques, or tau protein and neurofibrillary tangles, which produce reaction, which produce direct damage to the neurons, and you may have cell death. So there is a long time between the functional and the structural anatomical attack to the brain, which can last even decades. There are some evidences that speaks about 20 to 25 years. Why there are no symptoms? Well, there are no symptoms because there is a neuronal and cognitive reserve, and the brain is able to vicariate with this reserve all the loss of networks and neurons. But when this reserve is finished, the symptoms finally appear. Which is the technology most sensitive to synaptic sickness? The neurophysiological ones and the PET scan, but having differences in time discrimination and obviously in invasiveness and technological availability throughout the world and in cost. Let's see some examples of application of these modern techniques of EEG analysis. The analysis, for example, of the contribution of the different frequencies in terms of Reta method in a group of normal young people and old people. And it is clear also to an AFI that the amount of alpha activity in the different peaks of the alpha is quite different, and also the higher frequencies are quite less represented in the older brain, even if that brain is still healthy with respect to the young one. This is even more apparent when we compare a group of healthy elderly with a group of mild initial form of Alzheimer's disease. In this case, we have a clear-cut decrement of alpha activity in lower and upper alpha frequency rhythms, and the increase of the delta activity, so the lower type of frequencies. Genetics are playing an important role also in the generation of the EEG abnormalities. And it is known that today, still today, the strongest predictor of a genetic predisposition is still the epsilon-4 allele of the ApoE. The ApoE gene has 3 alleles, the epsilon-2, epsilon-3, and epsilon-4. Usually in the general population, epsilon-3 is the most common. And it is known that one allele of epsilon-4 increases of about 4 times the risk of developing Alzheimer. 2 epsilon-4 increased by 12 times the risk of developing Alzheimer. If we give a look to subject which we define mild cognitive impairment, and I will speak about more later on the meaning of this term, who are carriers of the epsilon-4 versus those who are non-carriers, again, you will see that in a completely identical neuropsychological and clinical condition, the carriers show much less amount of alpha-1 and alpha-2 EEG activity. If you make the same in the subject already being Alzheimer, this is clearly the same, much less alpha-1 and alpha-2 with respect to those who are non-carriers. So this genetic risk is also reflected on the genesis of the brain rhythms. In more recent years, the neuroscientific community started to speak about the connectome, which is the ability to measure the way that the neuronal assemblies, neuronal groups are speaking each other and are sharing information and during processing. And there are several methods. One method is the structural one, utilizing the diffusion tension imaging from MRI, so we can measure and see better and better year after year the fibers of connection. The functional imaging, the functional connectivity, which is a mainly statistical type of measure in which we look at the changes in time of the activity of the different brain areas, and whenever they change in a statistically related model, this means that they are working together. So we can track a measure of functional connectivity, and you see that the thickness of the connection is given by how more frequent is the transmission between, for example, an occipital and a temporal assembly of neurons, or between a parietal and occipital, and so on. And finally, we have the dynamic and effective one, which is able to follow the brain timing, which is in the order of a few tens of milliseconds. This time cannot be followed up by functional MRI, which is linked to the relaxation time of hemoglobin, which means 1 or 2 seconds at least. And moreover, the bold signal, which is at the base of the functional MRI imaging, is linked to any change of energy consumption. But we know that the brain is also speaking in a way that the energy consumption remains stable. For example, whenever you have a synchronization of the phase of 2 groups of neurons, they continue to discharge with the same frequency, they continue to consume the same amount of energy, but they are speaking each other. This kind of language cannot be followed up by functional MRI. So if we consider the brain as a container not only of oscillators, but also of structural networks, so groups of neurons with nodes, places in different brain areas, and connections, which are edges, we can consider them as networks again, and we can measure, utilizing the graph theory, the characteristic of this network architecture. And the graph theory is a mathematical theory which has several centuries of history, has been applied in several science fields, and not only on the brain. and gives us three different models of a connectivity which is fully ordered, a connectivity which is totally anarchistic, chaotic, a random one, and the so-called small world, which is considered the model which is closest to the real brain, in which, as you see, there are nodes with ordered connection, which are connected only to the adjacent ones, but some of them also have far connections, remote connections to other places. And also an if I can understand that if I make a lesion in one of these nodes, we have the final result of the lesion, which is only closer to the lesion side. But if we make the same lesion with the same volume here, the results, final results, will reflect not only to the nodes which are adjacent, but also to the nodes which are far away. For example, to this node or to this node. What happens if we, through this type of approach, we have a number of measurable parameters, not only the small world organization, which can be expressed with a number, but also the path length, so the length of the connections, the clustering coefficient, so the adjacency of the different assemblies one to the other, and many other parameters that we can measure. How do these parameters react to the normal brain aging? This is a study of some years ago in which we had three groups of young subjects, adult subjects, and elderly subjects, to which unfortunately I also belong now. And you may see that the path length characteristics, for example, are significantly different in three different rhythms, the delta, the theta, and the alpha 2, with respect to the three groups. And what comes out is that the elderly group has the shortest path length. It is like if the neuronal assemblies are becoming less and less able to speak to the far distant ones, and they only speak and receive information from the adjacent ones. It is something that happens in social sciences also for the elderly population. They usually speak more to the people close to them, they move less around, and they therefore speak less with persons at distance. What happens to the small world organization? These are three groups of subjects. A normal old group, so elderly with normal cognition, MCI, which is an intermediate condition between the normal, elderly, and demented people, and early demented people. This is the organization that we have through the different frequencies of the small world parameter in the EEG. And you see that the green, which is the normal group, and the red, which is the MCI, more or less show the same profile, On the contrary, there is a flat organization for the Alzheimer's. So there is a complete loss of the articulation of the small world organization throughout the different EEG frequencies when the brain is attacked by neurodegenerative mechanism. This is also very well related to the anatomy and the structure of this attack. We know that the first side, one of the first side of attack is the mesial temporal or medial temporal lobe and the hippocampus. And if we compare the characteristic of the small world and the other graph theory parameters with the volume of the hippocampus that can be measured directly on the MRI, there is a direct relationship. The more the volume is reducing, the more these parameters are affected. In more recent years, it became available a technique of a transcranial simulation which can be combined with EEG recording. So TMS EEG has been introduced in 1997 by Ilmoniemi et al. In Finland, and after that has been widespread all over the world. These are tracings that have been obtained from a 1020 recording EEG. This is a figure of 8 coil that is represented here during stimulation of the C3 windle, and then which means the left Rolandic brain area. This part of the trace is the pre-stimulus. Then we have the artifact, and you may see that before the stimulus there is a completely flat baseline, no activity at all. After the stimulus, after averaging some stimuli, we have a number of wavelets which are called transcranial evoked potentials, TEPs, which we can measure in latency, and we can also map out with a map. If we combine this with the neuronavigation system, we can know exactly where we are simulating, and we can follow in time the distribution of the transcranial evoked potentials during time. This is, I listen to a voice which is disturbing a little bit. Can you close your mic, please? Here we have TMS EEG applied to a group of healthy elderly, and see the organization of the stimulus of the evoked responses in EEG immediately after the stimulus and in different timing after the stimulus. And this is a group of subjects with initial forms of dementia. And if we apply statistics, we see that there is a significant difference at this time of about 80 milliseconds in the medial frontal lobes. So this P80, this wave, is considered a hallmark of the transcranial evoked potentials for this disease. If we apply the same approach to the MCI subject, and we make a subtraction of between the control group and the group with MCI, again we remain with a central to frontal statistically significant difference, peaking at about 80 milliseconds. So we are following up this type of approach because it seems that can represent a sort of biomarker. Along this way, recently, Giacomo Koch published in Brain the utilization of TMS, transcranial magnetic stimulation, as a magnetic treatment for this disease, stimulating mainly the precuneus. And these are the scores of a number of neuropsychological tests which have been followed up in time for the placebo group, which is a blue one, and the treated group with the real stimulation, which is a red one. And it is clear that the red group, the treated group, the group undergoing the real stimulation is doing much better in time compared to the one underlying sham placebo stimulation. So the transcranial stimulation not only can be a diagnostic tool, but might represent in the future also a new innovative approach for treatment and rehabilitation of cognitive deficits. Artificial intelligence is becoming a sort of might, or hopefully something useful in recent years. There are, as you know, a number of different algorithms which are now within the envelope of the artificial intelligence definition, and most of them have been applied already to the analysis of the EEG signals, and they are very promising because the EEG signal is really complex. It contains a number of information that the human eye can distinguish and discriminate in a very, very difficult and probably not possible way, in order to extract all the available information and to make it in an automatic and clinically valid way. This is a recent study in which we were utilizing a relatively large population of nearly 300 people, 120 healthy volunteers belonging to the old age, and 175 Alzheimer's disease patients. We were utilizing the
Excuse me, Professor.
Yeah.
Sorry, excuse me. Just to remind you that you have about 10 minutes left. That's
10
it.
more minutes. Okay,
10
more
more
than
minutes.
enough.
Yeah.
More than enough. Thank you. We were first utilizing the graph theory, extracting all the different parameters that I was speaking before, and in particular the small world. And then we were teaching to our algorithms and giving them and training them with the values of the graph theory parameters, and asking them just to classify, simply classify and to distinguish between the healthy old group from the early Alzheimer group, something that an ocular analysis of the EEG cannot do in any case. And these are the results. An ability and accuracy of 97% of correct discrimination. So the classification was very, very good, with a sensitivity of 95% and a specificity of 96% and a very high total accuracy. So this type of approach is quite promising. We need more validation, larger population, trial by trial discrimination, intersubject variability analysis, but the early approaches seems to be very good. Approximate entropy. As I was saying before the initiation of this lecture, I am not an informatic person. I am not a computer man. I am not an engineer. I am a neurologist, a clinical neurophysiologist, able to understand, but I am working with a large group of bioengineers and informatics and computer people. What I understood is that the concept of entropy is actually measuring the chaotic activity, the irregular activity, the non-ordered activity in a signal. And the EEG is a signal that is quite complex and contains a lot of entropic information, a lot of non-linear, a lot of chaotic, a lot of non-order activity, at least in an awake, healthy subject. The situation can change significantly in physiological conditions like during drowsiness or sleep at the different stages, or can change enormously in clinical conditions in which you have the loss of consciousness, for example, in comatose or vegetative stage patients. So we wanted to measure the different parameters of entropy. There are several parameters, and we were utilizing the approximate entropy analysis as an approach, trying to distinguish, for example, between the EEG of young age and old age healthy subjects. So we had 161 healthy subjects. Some of them, a larger group of them was relatively young, a larger group of them was relatively old, well distinguished in the age range. And the older brain, what we found out that we have higher values in the elderly group compared to the young one for the lower frequency bands, for the delta and theta, and a lower value for the beta 2 and gamma activities, which are those mainly connected with cognitive activities. So the older brain is not always more chaotic than the young ones. We were expecting something different, and the entropy analysis can really distinguish and classify correctly the old and the young brain. We are now trying to approach in the same way to distinguish between a pathological and physiological brain aging in different conditions. Again, you may see here the ROC curves that are quite, all of them very close to 94% of ability to distinguish between the two groups, utilizing the different types of artificial intelligence. The last part of my presentation is devoted to a huge public health problem. Dementia is a public health problem. We have in Italy today more than 1 million of demented people and about 800,000 people who are candidates to become demented. These candidates are belonging to a group which is called, as I told before, MCI, mild cognitive impairment. They have some neuropsychological deficits, but they are not yet demented. And the importance of this group is that only 50%, about 50 to 60% of them, when followed up in time, will become demented. All the remainings will never. They will remain a little bit amnestic with some neuropsychological problem, but completely autonomous in their daily life activities. So one of the main challenges today for every public health system throughout the world is to find out an instrument which is able to discriminate as early as possible within the group of the MCI that is represented in this square, who is the one that is going to become demented in the next 3 to 5 years and who is the one who will not. How high is the risk for each of these people to become demented in few years? Why? Because we can today intervene on some risk factors. For
Excuse
example,
me, Professor, you have 2 minutes. 2 minutes left.
Yeah, if you continue to interrupt me, I will have nothing. So let me finish that. I will do it in 2 minutes. So the problem is to have the instrument, and the instrument can be DAG. The instrument must be cheap, low cost, non-invasive, widely available, and obviously accurate. We made this kind of approach a few years ago. This is a publication in Annals of Neurology of 2018, in which we were combining the graph theory and Lorita approach to the EEG, together with the analysis of the ApoE genetic risk. Altogether, it costs about $100. The EEG alone is not performing quite well, even if now with the more recent artificial intelligence approaches, this curve is becoming better and better. But when combined with the ApoE information, then the ROC curve is becoming really interesting. So we can accurately identify 3 to 5 years before who is the MCI subject having a very high risk of becoming demented and who is not. The conclusion is that by utilizing neurophysiological techniques, by utilizing modern computerized approaches, by validating them in large population, making up multicentric type of studies, we can reach also an automatic approach of classification, which can be easily utilized also the general practitioners, so not specialized, not serving high aspect centers, and can be used with a very, very low cost in a non-invasive way and everywhere in both in high income, middle income, and low income countries. I terminate here, thanking all my collaborators, thanking again the organizer of this meeting, and hoping that the contribution I gave was of interest to all of you. Thank you.
Thank you very much, Professor Rossini, for such a great talk. Now, I want to give the floor to Yasseri Turia from the McGill University. He is working in disease progression and multi-scale neuron modeling in the human lifespan.
Okay, perfect. Thank you very much. Before starting, I would like to thank the organizing committee for the invitation to talk and the public for attending. In this talk, we are going to be discussing the work that we are doing for defining multifactorial disease progression models that can be used for biological defined diagnosis and treatment selection in neurodegeneration. rather than focusing on specific biomarkers or techniques, our group assumes an integrative approach in which we try to use all the available biological information for each patient. That usually includes from molecular markers or molecular type of data, including epigenomics, transcriptomics, proteomics, metabolomics, genomics, to multimodal. I was mentioning that our group focuses on an integrative approach that rather than using specific markers or techniques, tries to combine all the available data for each patient, including molecular omics, multimodal imaging, including PET, MRI, lifestyle information, clinical information, therapeutic information, and we integrate all the data using mathematical and computational disease progression models, and we believe that only by combining different sources of information, which we will be able to understand better the disease process and define personalized treatments. Of course, combining all this information is challenging, and for that we use different kind of computational techniques. There are two main families, or we focus on two main families of disease progression model. And the first one is empirical models, which are mostly based on machine learning techniques, which could be supervised or unsupervised, but which are good to make predictions. However, they not always offer a mechanistic or a biological explanation of the disease or the patient that we are studying. On the other hand, we have mechanistic brain models that focus on biological mechanisms rather than on making predictions. They can make predictions, but they don't necessarily offer high accuracy on the predictions because their purpose is to understand better the disease mechanisms. Just to put some examples, like about 3 years ago, we obtained post-mortem gene expression data for a population in the spectrum of Alzheimer's disease and Huntington's disease, and also blood gene expression data for Alzheimer's. All this data can be represented in the forms of the matrix, in which we have number of subjects by number of genes transcripts that could be about 40,000 or more. And then using a specific unsupervised machine learning techniques, we reduce that information, trying to maximize the contrast between the patients and the normal control subjects. This provides a set of a given number of principal components which are enriched in terms of representing disease patterns. We then assume that or use these components to represent each patient in or participant in a disease space in which proximity to the origin of that space will be reflecting or will be reflecting how close or far is a person to develop a neurodegenerative disorder. For example, we can see here that, and then we use specific techniques to identify trajectories on that space, and we identify 3 main trajectories that were reflecting or corresponding to a specific neurodegenerative process. As we can see here, these models identified a trajectory highly associated with Alzheimer's disease, other one with Huntington, and other one with normal aging. And we can see those persons that are closer to the center of the space, they are or they are not advancing a given disease process, while those that are far, they are closer or they already have developed dementia or the other symptoms associated with neurodegeneration. We then define it in a specific metric that we call gene expression pseudotime to calculate or estimate how far or close is the person from the origin. Remember that the origin represents the control, normal or clinically normal subjects. And then this measure, we confirmed that this measure was highly predictive of neuropathology in the brain and memory and cognitive decline in the participants. In other words, persons that presented the highest value of pseudotyme, that is a quantitative measure of how advanced is a patient or participant in the timeline to develop a neurodegenerative disorder. Those persons have advanced neuropathology in the brain and a stronger cognitive and behavioral deterioration. It is an empirical model, but still we can analyze, or we analyze the weights associated with each gene to go back and identify molecular pathways, genes, and cellular mechanisms that are highly enriched or highly informative about the disease process, and that are highly predictive in terms of if a participant will develop the disease or not. Recently, we extended that model to not only consider gene expression data, but also to aggregate molecular information from different levels, including epigenetics, transcriptomics, proteomics, and metabolomics. Again, we used advanced machine learning techniques to automatically identify trajectories on the disease, on the multi-omics disease space. And these trajectories are associated or supposedly representing putative Alzheimer's disease subtypes. Essentially, this approach allowed us to obtain two main indicators for each participant. One is an indicator of the disease subtype to which the person is belonging, and the other one is, again, a pseudotype measure that serves as a disease progression score reflecting how advanced is someone in the disease process and how close is to develop dementia, Alzheimer's dementia in this case. We then analyzed how the subjects in the subtypes were different, and we validated the subtypes in different ways, but also confirming that persons in different subtypes have different cellular alterations, even when all of them are developing Alzheimer's disease dementia. So this study represents or provided a multi-omics molecular taxonomy of the Alzheimer's disease, which reflected that the disease is highly complex, and that we actually should not be talking about one disease, but also about multiple or sub-disease variants that not only behave and develop in different ways, but that also might imply different therapeutic needs. And we will discuss after how to better select treatment for the different disease variants. We also have been working, as I mentioned, with multifactorial mechanistic brain models. We recently proposed a multifactorial causal model of brain reorganization intervention, in which we assume that changing a given biological factor at a given brain region, let's say increasing amyloid deposition or tau deposition, or increasing a gray matter atrophy at a given region, is a function, direct function of the multifactorial causal interactions between different biological factors. For example, how alterations in the vascular flow cause metabolic dysregulation, how those metabolic dysregulations cause functional hyperactivity or lower neuronal activity, how all those things together can impact amyloid deposition in the brain and vice versa, or tau increase in the brain. Plus, incoming effect from other brain regions. We know that all brain regions are connected, and what happens in one region will cause an abnormal change in other regions, so pathology change in other regions. Plus, if the person is doing physical exercise or not, which kind of diet the person is having, if the person is taking drugs or not, medical drugs or not. I will not enter in mathematical details, but I just want to say that these kind of models are used every day in many engineering applications that we use and in which we trust, including civil engineering, aerospace engineering, industrial engineering, mechanical engineering. So we use them to analyze how different brain properties or brain attributes or brain biological factors, like could be amyloid, the presence of tau, toxic proteins in the brain, amyloid proteins or gray matter atrophy, how they propagate, spread across time, across the different regions, but also how they interact in a causal way, as you can and we can see here. So this is the case for a person that is progressing in Alzheimer's disease, in terms of Alzheimer's disease, and we model how we feed the data and model and reconstructed how the brain of that person is changing since is 67 years old to 76 years old. We can see how there is a significant increase in amyloid tau deposition and in gray matter atrophy. We use this model to, in combination with genomics and transcriptomic information, to estimate or identify the current state of each patient, as we can see in this graph, and then estimate which would be the necessary brain inputs to conduce the person from the current disease state to a desired state that could be associated with improved clinical symptoms, or at least a state in which there is no further deterioration. And then that allows us to define or estimate individual therapeutic fingerprints that are numerical values reflecting which specific biological factors would be more convenient to target at the individual level. For example, we can see here that for patient number 1, the vascular or metabolic intervention would be more beneficial, while for patient number 2, an anti-amyloid and anti-tau intervention would be more beneficial. We also identify the molecular pathways associated with each type of optimum intervention according to the prediction of the models. And we verified in Parkinson's disease that those molecular pathways are highly predictive of response to levodopa treatment. That is one of the most common treatments in Parkinson's disease. So those patients that, or those molecular pathways that the model suggested that would be involved in levodopa, in response to levodopa, were actually the molecular pathways that we confirmed to be determined in response to levodopa in a population that received it. And the same, and that happens at the both genomic and transcriptomic level. In addition, we have extended the models to incorporate gene expression internally. So we have now whole brain generative models that in which are links between specific genes, let's say thousands of genes, and how they impact direct associations between different biological factors are estimated for each patient, which allow us to even create these disease road maps in which we can see which specific genes are modulating, let's say, increasing tau deposition in the brain, increasing or decreasing gray matter density. And also we can use this map to identify which specific genes we need to target to reduce or tau deposition, or to obtain a given desired effect at the multifactorial level and personalized level. We have also extended the models to incorporate neurotransmitter receptor densities, and this is work done by PhD in my lab in collaboration with Cal Series and Nicola Palomero from Jewish Institute in Germany. I don't have too much time to enter into detail, but the idea is very similar, is to model how the neurotransmitter receptors modulate causal interactions between different biological factors in the brain, and how the factors change together during disease progression, and how they will need to be targeted at the neuroreceptor level to conduce the person to a clinically normal state, or at least to a slow neurodegeneration. We have been also working to incorporate deepest molecular information, and we are now passing from not only including gene expression data, but also to include single cell RNA velocity, which is a new metric to characterize how the genes its activities in changing at a given time point. And it's a metric that reflects different aspects at the molecular level than just the gene expression or the other genetic information, the traditional genetic information. We know that implementing these models is challenging and they take a lot of time. So to facilitate the use in the clinic and in research, we have also created and shared an open access software that allows to incorporate or combine all the available information for each patient into mechanistic and empirical models that offer better biological explanations of the disease process at the individual level and potential treatments for each patient. I will also, I would finally like to thank my lab members and all the funding institutions. One quick note, I should mention that none of the results that I presented have been based on electrophysiological information, and none of them include EEG or MEG. And that's why one of the reasons or one of the purposes of this presentation is to motivate potential interaction with other researchers working with EEG and MEG, and we would be happy to discuss potential collaborations in terms of extending on models to incorporate electrophysiological measures. Thank you.
Thank you very much, Professor Yasser. Now I give the floor to the rest of the panelists so they can make questions about your presentation and Professor Rossini's presentation.
Hey,
Claudio?
And compliments to Professor Rossini for the impressive presentation. And my question is about how to manage the several quantitative measures for a clinical application, because I think that the approach is perfect for a clinical trial to test new drugs but in how the clinical hospitals can manage so many complex procedures for the computation of the measures.
may I read?
Yes.
Thank you, Claudio. Yeah, this is one of the major points, and what is needed by the neurophysiological community is to work together in order to collect sufficiently large and validated normative data, first of all, because the EEG, as you know, is changing in time. So we got to have a larger database of normative data with a clear pathologic cutoff on which then test our patients. This can be done today. It is not really too difficult. Harmonization of the recording methods is the second step. We need to have the same type of recording positioning, possibly positioning of electrodes, even if the 102019 electrode system provides sufficient information for a clinical application. And then we have what is probably the same problem for all the biomarkers, including the ones that Yasser has shown us, which is a homogeneous and harmonized clinical approach. In other words, whenever we have done multicentric clinical studies, as the Interceptor study in Italy or the artificial intelligence mind study in Europe, we realize immediately that even if we were thinking to make the same type of approach using the same neuropsychological test, classifying in the same way patients, this was not true. And we needed some days of harmonization for everything we were doing together in order to be sure that the definition of MCI was the same in the different centers. The recording system was the same in all the centers. And finally, you can really see homogeneous data, and you can really approach them in a proper clinical trial. At the very end, I really hope that artificial intelligence can help. But we have to train in the proper way. We have to supervise artificial intelligence in the proper way, in a clinically oriented way, in order not to have strange and sometimes wrong answers to the right questions, which might be even more dangerous than of not having any answer. So my hope is that clinical neurophysiology can be part of this early approach. And we have to consider that low cost is a major must. We cannot think of spending thousands of euros for each individual candidate, because this can be done in a clinical trial for a drug, because there is a company, a rich company behind that, but cannot be done on millions of people by a public health system.
Thank you very much.
Thank you, Claudio.
Thank you very much, Professor Rossini. Thank you very much, Claudio, for your questions. Let's give the floor to Maria Luisa.
Yes, I want to know about the EEG, because we are doing EEG for more than 30 years, and now this is very promising initiative. But I don't know if you can implement this easily, or you need more requirement. Also, because the data, the public data set usually don't include EEG. The ADNI don't have EEG. Many UK Biobank, etc., etc. What do you think about this?
Do you mean about applying my own models to EEG?
Yeah.
Yeah, so
It's
definitely
new data, no?
Yeah, so let's say that if we have access, we or any of the potential collaborations, to a rich EEG database, it would be straightforward to input the features extracted from the data into our software. So Paolo was mentioning features like network metrics and other features like from entropy, etc. Once that those things are calculated, so our software doesn't pre-process EEG data, but once the data is pre-processed by any other software or tools, all the features extracted from it can be incorporated, entered into software for applying any of the machine learning methods that we described. So there are specific ways. There is a tutorial that describes how to input the data. It can be in any way. We even are trying to implement ways to harmonize from across centers, although that is a critical issue, as Paolo mentioned. So data harmonization always need to be taken into account, and it's not trivial from the statistical point of view. But yeah, in summary, yes, we can try if there will be data available.
Okay, thank you.